Okay, so let us look at this subnuclear structure for our lesson today. Now, we do know that, okay, in a nucleus, we have 4 subnuclear structures. Okay, we have 4 subnuclear structures. So if you look at the previous recordings, you will know that these 4 subnuclear structures include nucleus, okay, chromatins, nuclear pore, as well as nuclear envelopes. So what are their functions basically? So first of all, let us look at the nucleus. So you look at this structure, okay, the structure inside the nucleus, you can see a very prominent structure. If you view it under the light microscope or under the microscope, you will be able to see a darkly stained regions in our nucleus, which is known as the nucleus. Okay, so the functions of nucleus we have discussed in the previous lesson. Okay, the function is to synthesize rRNA. Okay, synthesize rRNA. So R stand for ribosomal. Okay, the R stand for ribosomal. So it's a ribosomal RNA. So this ribosomal RNA later, when it move out from the nucleus into the cytoplasm, combine with the proteins to form the complete ribosome, which we'll discuss in the later of the subtopics. Okay, so this is the functions of nucleus. So in our syllabus, actually, you can write either way. Either you say synthesis, okay, involved in the synthesis of rRNA, or involved in the synthesis of a ribosome. Both correct. Okay. Then you can see that the next structures is a chromatins. Yesterday or the previous lesson, we talked about DNA. So enlarge this diagram. You can see that first you have the DNA. It's a double helix DNA. So this double helix DNA will be packed with, can you see that, wrap around the histone protein to form this thread-like structure. So this thread-like structure is known as the chromatins or chromatins fiber. So this chromatin fiber is very, very important because they contains the DNA, which is a hereditary unit in our cells. And in this DNA, part of the DNA or segment of DNA consists of the genes which code for polypeptides, which code for polypeptides. This polypeptide will fall into either proteins, enzyme, hormone, transport protein, which helps the cells to carry out the functions. Okay. Then next, you can see that this is the nuclear envelope. Okay, this is a nuclear envelope. So it's a double layer. Can you see that? Double layer. So that's why nucleus is a double membrane bound organelle. And nucleus is a double membrane bound organelle. And these membranes, they are selectively permeable. Okay, now very, very important, very, very important when you use the word permeability in this case. Now, I know that in some of the book, they use the word semi-permeable. Okay, now semi give us the idea of half permeable, which is not correct. Okay, because the membrane here, they are selective. They only allow a particular item to pass through, but they will block some item or some particle. Okay, so that's why we use the word selectively permeable. Okay, so selectively permeable, only some molecules will be able to pass through these selectively permeable membranes. Okay, so we term them as the nuclear envelope. So this nuclear envelope is perforated. Can you see that? You have the pore. So this perforated by what? Perforated by the nuclear pore or nuclear pore complex. So if you zoom it in, you can see that nuclear pore is not the empty space. Are you clear? It's not an empty space. So they have this what we call the central meshwork. Okay, allow a particular item to pass through. So for small molecule, they will be fully permeable. Small molecule have no issues to pass through. mRNA, no issue to pass through. The ribosomal RNA, the rRNA, no issues to pass through, but DNA is too large to pass through this nuclear pore. Okay, so we look at the first statement here. Double layer, Can I see that? Double layer. Separate the nucleoplasm from cytoplasm. So this is very important. So we do know that this must be the nuclear envelope because it's perforated with nuclear pore. So the first answer can join. Oh, sorry. To take. Okay, so connect to the nuclear envelopes. Okay, look at the second one. It regulates the passage of the molecule. So fully permeable to small molecule, but blocks the large molecules such as DNA from moving out of the nucleus. Okay, so this one must be nuclear pore. Okay, must be nuclear pore. Now, next. It is a prominent subnuclear structure. It's a very prominent subnuclear structure. Okay, so not bound by membrane and reside within the nuclear matrix or nucleoplasm. Involved in the manufacture of ribosomal subunits that combine to form ribosome. So it means that they are involved in the formations of the ribosome. So the DNA found in these regions encode for the ribosomal RNA. So synthesis of the rRNA. So this one must be nucleolus. Okay? So the last one here, they are complex between the eukaryotic DNA and the histone proteins, highlights DNA together with a histone protein. Okay, so DNA and histone protein, this one must be chromatins. Okay. So do you get the answer correct, everyone of you? Okay, so now here we do have some informations, extra information for you guys at this moment. There are two basic types of the chromatins. One is called euchromatin. Another one is called heterochromatin. Okay, what's the difference between euchromatin and these heterochromatins? When I say euchromatin, it involved in the transcribing RNA. Okay, so if you look at the structures of the nucleus, you can see that at particular regions, they are tightly coiled chromatin. Some part actually they are not tightly coiled chromatin. So in a tightly coiled chromatins here is termed as the heterochromatin. Not so tightly coiled regions here is known as the euchromatin. Okay, so what is the difference between this heterochromatin and euchromatin? Basically, it's like this. Now, imagine that's your clothes. Okay, if the clothes that you don't want to wear or seldom wear it, what we do? We pack them nicely and store it. Correct? You pack them into, let's say, for example, in a box. Okay, because okay, and keep it or store it. Correct? Not okay. But the clothes that you always want to wear it, you may hang them. Right? Pack them means nicely, but you hang them. Correct? Not loosely hang them, so easier for you to search. Correct? Not. So in our DNA, in our chromatins, we have two forms of chromatin. One type of chromatin, we always want to use them, so they are actively transcribed. So if they are actively use them, so it means that we do not need to pack them tightly. So this form of chromatin is called euchromatin. So it means that this one is okay. Let me write down. So it means that active transcribing. Are you clear? Active transcribing. So it means that we always use them. So we don't pack them in a box or pack them nicely or tightly. Heterochromatin is inactive. Okay, we don't pack them. Okay? So this one we're going to learn them in more detail when we come to the DNA, how we use the DNA to form or to code for the polypeptides. Okay, in your next semester. Okay, so again, guys, here is extra information at this moment. For your information at this moment only. So what we are going to look at is euchromatins and heterochromatin. Euchromatin involved in the transcribing RNA to synthesize polypeptide. Heterochromatin are transcriptionally inactive, contain highly repeated DNA sequence. So it means that we don't use them so much. Are you clear? So that's why we pack them into this very tightly coiled form, okay? But euchromatins is very active. We don't pack them very tightly. So again, when you look at this structure, you will realize that here is a very prominent structure. Can you see that? Okay, let me enlarge. So can you see a prominent structure? So this prominent structure is known as the nucleus, okay? Nucleus. And then you can see that some regions of the nucleoplasm tightly packed, not so tightly. Can you see that? Very tightly packed, not so tightly. So tightly packed is heterochromatin. This one is the euchromatin. So you can see that the nuclear envelope. So some part of the nuclear envelope appear as a pore. So this will be nuclear pore. Okay. So now can you label the diagram now? Okay, let me label it. So right now, so this one, oops, I think using other color better. So right now, guys, so this is the nucleus. Okay. You need to know how to identify the structure, yeah? How about this? This is either chromatin, but this one is the heterochromatin. Okay. We do have other chromatin as well, but this one is a euchromatin. Okay. Then you can see that's the membrane. So this one is termed as the nuclear envelopes. Okay, nuclear envelopes. And you may see that's the pore. Okay, you may see a bit of pore here and there. So this one is the nuclear pore. Okay. So please label. So once again, you should know now how to identify it, okay? So if you do not, or you forget which one, heterochromatin or euchromatin at this moment is not so crucial. Just put it as chromatin at this moment. Are you clear? But when we go for this protein synthesis, then we need to know heterochromatins and euchromatins, okay? So nuclear envelope, nucleus, very prominent structures, and together with nuclear pore. So this one, we view it under the TEM, transmission electron microscope, which we're going to learn in detail when we come to the electron microscope. So if you look at the TEM microscope, okay, or TEM, then you can see that it's a 2D. They form 2D diagrams, okay, or picture. But if you use a scanning electron microscope, we scan the surface, it will give us a 3-dimensional. So you can see that nuclear pore with a complex there, or nuclear pore complex. Okay, so point to one nuclear pore, and write down this is a nuclear pore. Because when we use a SEM or scanning electron microscope, we only focus at the surface, so you can't see the nucleus at this moment, right? Because we look at the surface, okay, at the surface of the nucleus. Okay, so with this, I have done. Okay, I have done for the nucleus part.